Welcome, everybody. We have about a minute to start. This is a human space exploration session. My name is Ignacy Górecki, and I will be leading this session. Sadly, we are coming to an end of our conference. We hope that you enjoyed those 3 days with us. Certainly we did. I see that our audience is slowly growing. And I can see that our first speaker is ready. Arkadiusz Kolodziej is a medical student, currently at a research position at the University of Virginia. We'll talk about outsleeping the space travel. So also a quick reminder to speakers, please stick to the 15 minutes time slot prepared for you. I'll let you know about 3 or 2 minutes before the end of your time. And Arek, without any further ado, the floor is yours.
Thank you. If you like sci-fi movies, you're probably familiar with the idea of hibernation. If not, I can tell you that hibernation would prepare us for deep space exploration. It could literally save our life in space, or at least extend it. My name is Arkadiusz Kolodziej, and I am a final year medical student and a visiting graduate student at the University of Virginia. Today, I will try to show you why and how this sci-fi movies idea might be helpful in the case of human space exploration. Let's start with defining what hibernation and torpor really are. Torpor is quite weird, peculiar biological condition. It occurs in mammals such as squirrels, hamsters, mice, but also bigger mammals like bears. And it is characterized by a reduction in metabolic rate that causes a progressive decrease in body temperature. The term torpor usually refers to a single bout of metabolic suppression that lasts from a few hours to a few days. On the other hand, hibernation is a sequence of torpor bouts, and it is often interrupted by brief interbout arousals that can last around 24 hours. Hibernation itself can last anywhere from a period of days to weeks to even months, depending on the species. The basic aim of this state is to induce a reduction in metabolic rate that could allow hibernators to survive in the conditions with some problems like reduced access to food or water. As you can see in this table, the duration of hibernation varies among different species. What is more, the decrease in body temperature in these hypometabolic state can either be extreme, such as in the Arctic ground squirrel, that can reach a below zero temperature, or mild, such like in the case of bear, when the temperature can drop rarely below 30 degrees Celsius. Seasonal hibernators, such as the Arctic ground squirrel, have a strong drive to enter hibernation before the arrival of winter. Which is interesting is that the body of these animals pre-adapts to the hibernation season, reducing, for instance, other functions like they can reduce testicular volume or thyroid activity. These changes suggest that some kind of genetic regulation of this behavior might be at work. So it is reasonable to think that there's a single underlying mechanism that reduces metabolic rate. However, it is not well known at this moment. A complete understanding of the molecular mechanism that triggers torpor would be of great help in the quest to develop a procedure to induce this state in humans. But why would we even want that? Many of the changes encountered by the body during hibernation could in fact be extremely helpful for humans in a long-term space exploration mission. Among the most beneficial features of hibernation, I could highlight reduced food and water requirement, reduced waste production, preservation of muscle strength and bone structure, and enhanced radioprotection. The last one seems the most interesting to me because to date, we do not have any effective countermeasure against the radiation in deep space. While, in case of food or water, we would probably take more food with us or just produce some on site. Let's talk about physiological adaptations in hypermetabolic states, in particular parts of our bodies. I start with the brain because, first of all, this is my favorite organ, and second of all, it shows the most drastic changes during torpor. In squirrels, synaptic spines were shown to be largely reabsorbed in many portions of the cerebral cortex, as well as in the hypothalamus. The reduction in the number of synapses may be an adaptive response that aims to reduce the high energy expenditure of the brain. Synapses are then reformed very quickly after the return to normothermia, which is visible in this chart. In the last row, we can see changes in temperatures and corresponded to them changes in synapses in the first 3 rows. At this moment, little is known regarding the characteristic of the reformed synapses compared with the reabsorbed ones. Are they in the same location? Do they have the same strength? This very peculiar event could be an interesting model to study synaptic plasticity. But the time needed for the rewiring of the brain must be taken into account, especially in the event of emergency arousal, since the recovery to full cognitive capacity will probably take some time. Besides the reduction in the number of synapses, neurons also present changes in some of their key cellular structures. In particular, tau protein becomes hyperphosphorylated. You can see that in this figure. Let's focus on this postsynaptic side, so the greeniest part of this figure. On the top, you can see in which part of the hibernation cycle we are. So we're talking about torpor and short arousals. And the tau protein is phosphorylated because it glows red here. This molecular change resembles the one key change that is associated with Alzheimer's disease. The purpose of this phenomenon is not known at the moment. We don't know if it served to protect some structure of the neuronal cytoskeleton from damage, or maybe it promotes a further reduction in energy consumption because it lowers the axonal transport. We don't know that. What is clear is that shortly after the arousal, phosphorylated tau protein is cleared from the brain, which you can see in the figure in the parts after the torpor state, so in long arousal. It is certainly suggested that in torpor and synthetic torpor, the brain can be seen as diving into quickly reversible dementia. One more trivia about the hibernated brain from my field of study that I do now. You may think that torpor or hibernation is just like sleep. However, it was proven in EEG recordings that it is not. Frequencies presented during hibernation differ significantly from all the known sleep stages, and some authors call this state slow wakefulness. Let's briefly mention some of the other parts of our body. The gastrointestinal tract is affected by hibernation in a variety of ways, as you can see in this figure. But I would like to focus on the most important parts, highlighted with the red frames. So it is, in fact, mucosal size and protein content, as well as villus height is reduced during hibernation. Interestingly, also, barrier becomes generally leakier, since the number of gap junctions decreases too. And under normal circumstances, that could be problematic because it would expose organism to infection. But during hibernation, an increase in the lymphocytes occurs in the GI tract that possibly prevents from microbial invasion. Speaking of bacteria, the microbiota is also significantly changed by hibernation. Intestinal microbes adaptively reorganize their configuration during torpor. The number of bacteria strains that could survive without external nutrients increases, and these bacteria eat mostly carbs found in mucosa. What is more, changes in microbiota were also shown to induce changes in metabolism when transplanted, possibly playing a role in guiding or favoring the metabolic suppression in hibernation. The possible utilization of tailored probiotics to modify the gut ecosystem during therapeutic hypothermia can also be taken into consideration as a new therapeutic strategy. How the locomotor system behaves in the state of hibernation is extremely interesting, because we do know that disuse atrophy of the skeletal muscle is consequential to the reduced mechanical load in conditions such as prolonged bed rest or microgravity environment in space. However, there are a lot of mechanisms that could influence the preservation of muscles and bones during hibernation. As you can see in this figure, they can be divided into mechanisms associated with hyper-metabolic state, intracellular mechanisms, and systemic mechanisms. Hibernating animals are subject to extended periods of muscle inactivity and fasting, but they do not experience disuse atrophy. Moreover, the functionality of the skeletal muscle is preserved both in small hibernators, such as the squirrel, and in the larger, such as the bear. Similar to skeletal muscle, multiple reports show that bone structure of hibernators is also maintained during hibernation. And now, let's talk about radiation. To my mind, cosmic radiation represents the biggest threat to the health of astronauts during long space flights. Most of the studies investigated the effects of radiation on hibernators showed that this hypermetabolic state increased the resistance to radiation and probability of survival, which is visible in this chart. However, the studies conducted on Earth have so far used X-rays or gamma ray irradiation. No study has yet investigated the radioresistance of hibernators to the charged particle component of cosmic rays, which plays a major role in radiation exposure in the deep space. Here you can see even more possible mechanisms of the hibernation and their effect on the problems that we actually already do face in space. Overall, the understanding of the physiology of hibernation could be very useful to make human exploration of the solar system possible, but could also be exploited to improve the treatment or outcome of numerous clinical conditions that are treated with radiation on the planet Earth. Jumping into clinical stuff, I'd like to highlight some cases of hypothermia in humans, because patients surviving deep accidental hypothermia may help hibernation research in understanding the role played by hypothermia during torpor. Today, it is not difficult to find records of people that survived many hours with no heartbeat in circumstances in which they were exposed to severe cold. From the 17th century case of Anne Greene, a lady sentenced to death by hanging that survived the execution and was revived by physicians, to the much more recent case of Anne Bagenholm, probably one of the most famous cases of this kind. Another observation that supports the hypothesis that humans could enter torpor is found in the measurements of metabolic rate in human fetuses. Fetuses are exposed to the high body temperature of the mother and to lower oxygen tension than adults, conditions that could both influence metabolic rate. So, do we have what it takes to hibernate? So the scientists think that the ability to hibernate arose with the first ancestral mammal, a fury tree-dwelling creature that lived 65 million years ago, and eventually gave rise to the most modern mammals, including humans. If they're right, the key to hibernation may be hidden in the human genome. Indeed, according to fossil experts, evidence from bones found in Spain in 2020 suggests that our hominid ancestors may have dealt with extreme cold hundreds of thousands of years ago by sleeping through the winter. In 2013, the first successful procedure to induce a reversible state mimicking natural hibernation in a non-hibernating animal was discovered, which was proposed to be called synthetic torpor. It was done in a quite invasive way by injecting muscimol, which is the agonist of GABA receptors. However, synthetic torpor could be induced also in other ways. At the moment, 4 procedures have been shown to be effective. The first one that I mentioned, the second one, which is the activation of adenosine A1 receptors in the brain that influence sleep and wake behavior, and a cocktail of drugs, including xenon, and the activation of hypothalamic Q neurons that control the body temperature. The important feature of the torpor is the ability of the animals to arouse spontaneously from the hypermetabolic state. And so far, this spontaneous arousal was described for all these 4 procedures. All these methods require more translational research before being used for space exploration, but the objective to be pursued is not now clear, and this ambitious goal is no longer out of reach. At the beginning, I mentioned sci-fi movies. Here's what we could see in sci-fi movies. Now, when you're familiar with the topic of hibernation, do you think that we could achieve something similar to that? Because I think we could. You may think that I'm crazy because of that, but I'm happy with that because I'm not the only one. This is the real thing from NASA Innovative Advanced Concepts. I'm not even kidding. There are groups of scientists that work on that topic now, and this figure presents how it could look like. I hope that I've warmed your imagination and kinda interested you with the topic. But let me say one more thing. If we want to hibernate humans, it is extremely essential to find safe and non-invasive way to do that. Therefore, as a summary, we ended up with some critical questions that still remain unanswered. Does hibernation for radiation protection also apply to the radiation that we can find in galactic cosmic rays or energetic solar particle events? And how do we balance the torpor and arousal cycle in synthetic hibernation? And also, what are the side effects of this state? We should work on that topic to answer these questions eventually and prepare for the deep space exploration, because we will do it one day, I'm sure. Let's do it safely then, and maybe a little sleepy. Thank you for your attention.
Thank you, Arek, for this presentation. I really enjoyed those presentations where science meets with science fiction. We do have one question for you in the chat box from Martin Wysiecki. Would we dream at the time? And so, will we age more slowly or maybe not at all?
So it is not really, as I said, EEG recordings of the animals that were hibernating, they shown that this state is not similar to normal like sleep patterns. So it's difficult to say if you would really dream or not at this point.
And what about life expectancy? Would there be any difference?
So probably yes, because like life expectancy in space, probably yes, because as I showed you in, I can actually, if I'm still screening, there was this, yeah, this is a mortality rate, and so survival
I'm afraid
animals.
we can't see your screen right now.
Oh, you can see my screen. Okay.
No.
So I showed you that mortality rate in animals that were exposed to radiation was lower when they were hibernating. So it is possible that that we will live longer because we will be more protected. But also the aging process, it was found to be kind of affected by this state of hibernation. But at this point, it's difficult to have any conclusions on that because more research is needed.
Okay, I have one more question because we touched a little bit about immune system, and it is one of the ways that we can fight with cancer development. So, is there anything? How much is immune system affected by this state?
Mm-hmm. That's also a really interesting part of that, because there are some changes in immunology in space, and especially in in hibernation too. And so the main one is leukopenia, so the reduction in number of circulating white cells. But in state of hibernation, these cells do not die, but most of them travel to organs like liver or spleen or intestines that I mentioned. And they can go back to bloodstream after arousal. And the question now is if this state makes hibernators more susceptible to infectious diseases. And in fact, the answer is no, because hibernators do not die from infectious diseases more than like other animals from the population. But the exact mechanism of this kind of protection is not well known. But probably it's because of these white cells that stay in this kind of reservoir organs that I mentioned.
Okay, thank you, Arek. I can't wait to learn more about hibernation.